Alright, so we'll be reviewing the cell membrane today and just going over its functions and the different components of it. Now, at a first glance, it may look really complicated just looking at this, all these different names, but it really isn't that hard actually, and it'll become much easier when explained a little better. So first, let's just talk about its function. So function. So the function of the I kind of jumped ahead of myself there, but the function of the cell membrane is that it is a biological membrane enclosing and separating membrane that acts as a selective barrier. So that's what it basically does. It basically acts as a barrier for what enters and exits a cell, essentially. Now, it does this in many ways. That's what these components are for, and that's exactly what we're going to get into right now. These components are called, well, membrane components. That's how I remember them. And the first thing to note is that, let's just title this components. This is a very extensive area, but the ones that you should know are, firstly, you have this little thing that looks like some kind of bacteria or something, but it's not. This is what you call the phospholipid. Wait 1 second. This is not a bilayer. It has to be like this for it to be a bilayer. Okay, my proportions are very off there. I'm sorry, but this is what we call, I'm just gonna label this as well so that it's easier to understand. Phospholipid bilayer. So I only drew 3 here just to explain a little better. So they have a head here. This is what we call the head, but we call it a hydrophilic head. And we call it that because this part of the phospholipid bilayer faces the water. And so as such, it's hydrophilic. Philic meaning water loving. Hydrophilic. So when you say, this is a pretty common word that you use in like day-by-day life. You know, like philic means, you know, like to have a liking towards something. So I just think of it as water loving, pretty straightforward. So the heads are water loving hydrophilic heads, and they're made of phosphate. So I'm just gonna move this along a little bit so that I can just write it before. So the heads are made of phosphate, and so they are hydrophilic. And these things, this is what we call the tail. And this is made of fatty acids. Fatty acid tail. Hydrophobic. So hydrophobic again, another terminology. This is also, well, pretty straightforward, used day by day as well. Phobic means to be afraid. Well, but it's not actually afraid. It's more so just like it doesn't like it. It's water hating or water disliking, or it doesn't like water basically. And this arrangement actually really complements this hydrophobic arrangement, because as you can see, so this is the, let's assume that this is the cytosol, so like the cell membrane or whatever, right? Sorry, the cell environment. This part is facing the water, and so as such it's hydrophilic. And these this tail part is on the inside, and it's just like huddled up on the interior away from the water. So it's arranged that way, and it's done this way on both sides. And as such, it creates something called a bilayer, which means it has two layers of this going on, right? So it's a pretty fascinating model if you really think about it. You know how cool it is that we have something that you know, molds itself around what it likes and dislikes, and how that's really important to its function, which we're going to get into really soon. You know, so that's the first aspect. And on this bigger model, this is what we're referring to. This is the heads, the phosphate heads, and these are the fatty acid tails. And look at the arrangement here, similar to what we did. And it says over here that this is the hydrophobic core, because these are away from the water and they're all hydrophobic. And these ones on top, which are facing the water, are hydrophilic. I think that's meant to be hydrophilic polar heads. All right. And these tails are made of fatty acids, like we said, fatty acids. All right, let's now move on to the next component of this, which is something we call cholesterol. Now, you've probably heard of cholesterol. It's used quite a lot in day-by-day conversation, but the main function of cholesterol, I don't know if it's on this diagram that we have here. It isn't, but it's present. It's here somewhere. It's in between the phospholipids, and its function is to restrict the lateral movement of phospholipids. And also, it stabilizes interactions between phospholipids. Alright, so firstly, the restricting the lateral movements of phospholipids. I didn't mention this, but it's actually quite an important thing. The phospholipids are consistently moving, and we refer to this as the fluid mosaic model. We'll get into that after we finish with all the components, because it makes a lot more sense looking at it holistically in the end rather than looking at it one by each component, you know. So what you need to know right now is that the phospholipids are fluid, so they move, right? And oftentimes they can move a little too much, right? And that's not good for our cell membrane because it can cause it to collapse, right? So it doesn't perform its function anymore. Anything can come, anything can go out of your cell. Not optimal for survival, right? So as a result, we have cholesterol present in between, like here, here, here. What it does is that it restricts the lateral movement of these phospholipids. So lateral means and what cholesterol does is it's like, okay, like these guys are trying to move this way, these guys are trying to move this way. It's like, nah, like you're gonna stay where you're at, and like just preventing that movement so that the membrane doesn't break. It's really quite important. Which is why oftentimes you'll hear people talk about how it's not so good for you, this, that, and the other, and that is true because excess of it isn't good for you, because it's also involved in other functions in the liver, which obviously aren't so great if you have too much. But it is also like quite crucial in your cell membrane. So obviously at a moderate amount, it's really important. So that's what cholesterol does. Let's now move on to the next component that we have, which is something we call a glycoprotein. Let me just write that down, glycoprotein. And in its name, you can really tell, glyco means something that comes from a carb, and protein, well, that's a protein, right? So a glycoprotein is a carbohydrate that has been modified to act, that has been modified to, to contain a protein on its surface. All right, so that's typically what it is. They basically act as receptors usually, so we can just mention that, receptors, and we can see them on the diagram that we have here. That's the glycoprotein, so that's the protein aspect, and that is the carb aspect. All right, let's now go over what a glycolipid is. Also really straightforward in its name, glycolipid. So carbohydrate that has been modified to contain a lipid. But in this case, however, the lipid is already present over here in the form of the bilayer, right? Phospholipid bilayer. So the carb just binds itself, you know. Oh, and sorry, previously I did mention that this is the protein, but actually it's the other way. I'm sorry for that little overlook on my part. All right, let's move on now to the next part of this, which are the types of proteins. These are also components, so we're just gonna keep writing them. So there's two types. There's integral proteins. So firstly, let's talk about integral proteins a little more. I'm gonna bring the model which I made over here. Wow, so perfect. All right, now let's talk about what an integral protein is. So integral proteins are basically, this is what it is. They span the entire length of the bilayer, so they're like pretty long, and oftentimes their functions are, well, they sometimes act as co-transporters, sometimes they act as channels. So in the case of channels, the center is usually hollow and surrounded by water and allows the ions or whatever it is to move across. In the cases of co-transporters, it opens and it closes and also encompasses the whole thing, you know. A whole bunch of functions, really, really important. Good to know, have to know actually. So that's integral proteins. And if you had to look at on the diagram, see, here's an integral protein. It's going all the way across. This even has a receptor on its surface. So it recepts, it firstly gets, it detects which ion that is coming in, and then allows it, because these are selective, they don't really just open for anything. Some of them do, but some of them don't, so that's something to know as well. And again, this is also integral protein, this as well. They're just like all across the cell membrane, and they're really important for allowing cell transport. Let's now move on to peripheral proteins, or the other kind. peripheroproteins. And once again, I'm gonna borrow my model from up. Can't be bothered to draw that again. Duplicate. Now let's bring this down. All right. Let's try. Well, actually, let's move it a little further down. Okay, so these proteins are usually on the surface, so they're like this. So they're on the surface, and we can look at examples of this on this here. These are peripheral proteins. They're usually on the surface. So that's the difference between integral and peripheral proteins. Integral means that it's integrated into the cell membrane, and peripheral means that it's a little far. But that's the two types of proteins that you should know. I think I should write it down. Proteins spanning the length of the phosphorate, of the cell membrane. And this one, proteins spanning, well, it doesn't really span anything, proteins on the surface of cell membrane. Alright, so that's that. Now we have one more component to go through, and then we're going to go over the fluid mosaic model, and then we're pretty much done for this video. The last component to know is, this is really a subcomponent, but it's good to know. Well, it's a kind of carb, so we already spoke about carbs before, but we're going to just refer to it again. It's called a glycocalyx, and these are membrane carbohydrates that extend from the outer surface. These also usually they're basically what integral proteins are, but carb form, so they span the entire length, and they usually act as receptors, and they usually work in conjunction with the glycoproteins and stuff like that. So that's ideally where you'd see it. It's quite important as well, for different kinds of recepting and other functions as well. So that's all of the components that you should know. Let's now quickly go over the fluid mosaic model, and then we'll do a small little bit on semi-permeability, just referring to that a little more, because that's actually quite an important aspect of it, because not everything can go through, and things can go through, but it depends on what their their methods of entry. Fluid mosaic model. All right, so a fluid mosaic model is well, let's break this down. Let's first go over fluid. Fluid means that it's moving, right? Moving model, and that's actually true. In this, obviously it's a still image, so you can't really tell, but they're consistently moving laterally. Like, it's just a thing, they're constantly moving. And it's called a mosaic because what is a mosaic? It has a bunch of components to it. It's really studded. So the reason we call it that is because it's studded with components. And as we refer to all these components, you can see here, it does kind of look like a mosaic. A bunch of different things, this here, your glycoproteins, your cholesterol, your integral proteins, your glycolipids, everything is studied all across the cell membrane. So it kind of looks like a mosaic, which is why we call it the fluid mosaic model. And this image is what we refer to when we say, oh, it's a fluid mosaic model, because it's a mosaic, but it's not moving, but it's still fluid. Like, we know that because of all the way that it is. Let's just say that cell membrane is referred to as fluid mosaic model because of its movement and the fact that it is studded with various components. Now, you can obviously mention them as an example, the components and things, but I'm just going to leave it there. Let's now quickly go over semi-permeability and the different kinds of molecules that can get through, and like their methods of entry and things like that, just so we have an idea. Of course, we'll go into more detail on this later. It's more like an entry-level review of this. So the different kinds, firstly, we have four that are like quite important to mention. Firstly, we have hydrophobic molecules. So as it says in the name, hydrophobic, water-fearing or water-hating. And these ones, molecules that readily pass through bilayer. And the reason that they go through readily is because they can record, like there's nothing stopping them basically, and there's no water component there. The inside is all fatty acid, so it doesn't have any water. So since they fear water, they can get through really quickly. And there's water on the surface, which is why it needs to escape that. and starts moving across, if that helps. And so examples of this could be, well, oxygen, H2, and also under this are like really small like things, really small molecules which can get through, like without really even needing a way to get through. They're really tiny, like oxygen and water. And steroids are your examples of hydrophobic molecules, which well, they can get through because they are really they can get through really quickly through the lipid aspect of the phospholipid bilayer. Let's now talk about the oh, sorry, I mentioned water here. That's not true. It's actually carbon dioxide. Sorry, that's my bad. What is actually part of this next section? This is what we call small uncharged polar molecules. That makes more sense. I knew that I had a different section for it, and I did. So these are the ones that I just mentioned. Some molecules that can pass through without needing a transport protein. And these are just so tiny and stuff, and they're also uncharged, so they don't really need anything to get them through. So examples are water, examples are urea, ethanol, so alcohol, and glycerol as well. So they're so tiny and so uncharged, they can just get through. You don't need to really even have anything. I'm just going to continue here because I don't really want to take another page. Other ones are large uncharged polar molecules. So they're basically they're different, and they get through very difficultly. Actually, they have a very low membrane permeability. So they really need the channel proteins and all that to get through, or like different methods of getting in, such as secondary transport mechanisms and things like that have very low membrane permeability. And so examples of this classic example is glucose. And then lastly we have ions. And ions are just straight up not permeable. Ions are not permeable. And so for them to get through, they definitely need the channel proteins and all that thing to get past. So let's just write some examples: Na+, Ca2+, H+, K+, so on and so forth, chloride, etc. There's so many. So that's a quick review of semi-permeability, cell membrane, and fluid mosaic model. Hope that helped.